Hello, my name is Robin Dewsbury. I'm the author of the CheckIron website. We started CheckIron 5 years ago, and I'm going to tell you in this video why we did the work, what's in it for anyone who's got a consumer DNA test results, and about our many new research discoveries and what to expect in the future. So what is CheckIron? CheckIron.com is a free and anonymous software web-based tool which analyzes your raw DNA data from providers such as Ancestry or 23andMe and many more. CheckIron is a citizen science, data science project. It makes no money and never will. It tells you your risks of suffering from hereditary hemochromatosis and related inflammatory diseases. It even goes as far as estimating the age at which it thinks you could get sick to a statistically determined accuracy of about plus or minus 12 years. We predict the age of suffering. You still get many people saying genetics cannot predict when someone will get ill. We challenge this idea. We believe CheckIron can be used as a prevention tool. Hemochromatosis can be treated early using phlebotomy, and doing this results in prevention of illness. But we need more data to definitely prove this. So what is hereditary hemochromatosis? It's a genetic fault which causes iron overload, where the body is unable to control the amount of iron being ingested from your food. Iron cannot be excreted through the kidneys like other poisons, and iron overload therefore accumulates as we get older. The only real effective way to remove iron is to remove blood from the body. There are different types of hemochromatosis based on different mutations and 5 different genes, and the 5 different gene hemochromatosis types are called hemochromatosis 1 all the way to hemochromatosis 5. Some are rare, but others are ignored by medicine because they're thought to be rare, but they're not. There is something called classic hemochromatosis, which is a double mutation of the C282Y HFE gene. It is not the most common form of iron overload, yet there are some older doctors who still claim it's the only form which is important. And this is absolutely not true. Hemochromatosis is the most prevalent genetic disease for people of Northern European descent. It affects 1 in 300 Americans, 1 in 150 people in England and Wales, 1 in 113 people in Scotland, and as many as 1 in 10 in Northern Ireland. In the Republic of Ireland, it's 1 in 83. And then from Scandinavia and northern shores of Europe, but most are unaware that they have it. Separately, even high iron is unhealthy for people with or without the genetic disease, and the number of people with this are similar to those with iron deficient anemia. Awareness of this condition is non-existent, also and severely needed. The result is, for those with the gene's disease of iron overload, they get cirrhosis of the liver, liver cancer, heart disease, arthritis, Alzheimer's, and Parkinson's. Many people die from these secondary diseases without anyone realizing the ultimate cause was actually genetic iron overload. Clearly, hemochromatosis is only responsible for some small proportion of these diseases, but for those that do have iron overload, The treatment is simple and relatively risk-free, just regular blood removal, and as a result, many lives could be saved. So who am I? I'm the developer of the CheckIron website. I have a PhD in physics and in particular doing experimental research. I've spent 35 years working in computers and mobile phones, mostly specialized data science, computing simulation, mobile apps, and artificial intelligence. I ran several startups during my career at the end. CheckIron is a research data science project, and as I explained, we make no money and never will. I discovered I had hemochromatosis when I was 60, just from my DNA. And I was amazed no one used DNA to discover hemochromatosis at that stage. So I started CheckIron in 2019, just 5 years ago. Now, before we actually show you our own data, I'm going to show you some published data from major studies where large numbers of people have been involved. So this table actually shows numbers from two different scientific papers. The first is from a study of 9,000 inhabitants of Copenhagen in Denmark over a 20-year period. And the second paper is genetic analysis from the US of the prevalence of particular mutations. Now, the other thing to say about this table, it's only for hemochromatosis type 1, which is the most common type, and it's only showing two different mutations where actually hemochromatosis type 1 has three different mutations. But the reason why we only showed two is because most people are only interested in two, because they're the most common, and the third one is often not included. What we can see here is that classic hemochromatosis, which involves C282Y as a double mutation, and is in fact the disease I have, comes out as being 1 in 379 people in the US population. As I mentioned earlier, this double C282Y is supposed to be the most common version. And in fact, as I said earlier, a lot of older doctors think it's the only version that affects people. But have a look at right at the bottom here, the H63D stroke WT, which is WT means wild type, and in fact what it means is a single mutation of H63D. This is actually quite a common mutation, so you have a high prevalence but a low penetration. It's actually only 2%. But if you multiply prevalence by penetrance, you get the number of people. And this comes out as 1 in 279. Now, that's actually more often than the classic hemochromatosis. And that's what I meant is many doctors will say carriers never get it, but here's some calculation which shows it's more common than classic hemochromatosis. Now, please note, this is not diagnosed people. This is suffering people. This is people with blood tests that show they likely have iron overload, but not that they've been diagnosed by a doctor. And the reality is most carriers are never diagnosed because they incorrectly think that carriers can't get it. And we'll talk about that a bit later, why that is. Now this next graph is even more interesting. It shows the 9,000 people who are studied from Copenhagen over 20 years. Now, the point about these 9,000 people is they were just ordinary people. Some had hemochromatosis and the majority didn't. But what was looked at was the amount of ferritin in their blood. This is independent of which genes they had. And it actually shows that ferritin is actually quite has a bad effect on the age at which a person will die. So if you look, the dotted line, which collapses at around age 30 and then remains low, is people dying from 30 onwards all the way up to 90. And this is for people with ferritin levels over 600. Now this, by many doctors, isn't considered enough to cause iron overload. Many believe you have to have a ferritin of 1,000. But I think increasingly people are accepting that lower numbers, anything above 300, is actually bad for you. And this study actually explains why it is, because at over 600, you'd expect the median age of someone to die at around 55. And when you think of it, when I discovered that I had hemochromatosis when I was 60 and my ferritin level was over 1,000, I immediately realized, according to this graph, I should have died 5 years earlier. But that's the nature of statistics. Unfortunately, I discovered it and was got cured before I did die. Now let's look at our own data. So we are members of a number of hemochromatosis groups on Facebook, and it's those users who are interested to find out more about their genetics. And we offer them for free. In fact, we offer any members of the public for free, the ability to upload their DNA data from one of these commercial websites such as Ancestry or 23andMe. So over the last 5 years, we've had nearly 2,600 people upload their data. And of those, if you think they're coming from a position of probably already suffering from hemochromatosis, 1,800 of those users, that's 70%, had the hemochromatosis genetics, not just type 1, but the other types as well. Now, one of the questions we ask as we show people their results is what age they were when they got sick, when they got symptoms, and if they hadn't got symptoms at this stage, we ask them what age they are. And 847 have told us their age when they got symptoms. This is actually a bit disappointing, but we'll try and persuade more to do so when they understand the reason why we need to know this data. Of those, 158 said they didn't have any symptoms yet, and the remainder, which was 56%, didn't answer the question, which is a bit disappointing. Now, in terms of what we're looking for, we're looking at nearly 300 mutations which are related to the iron metabolism and the immune system, because one of our discoveries is the immune system is heavily involved in the iron metabolism. And of those mutations, there's actually 106 genes. Now let's get into our actual data and our discoveries. One of the things that I've already implied is that type 1 isn't the only type of hemochromatosis which is common. It's still the most common, but one of our discoveries is that type 3 is nearly as common as type 1, and in fact 19% of our users had both type 3 and type 1 at the same time. This is something that isn't reported by anybody else. And the reason why it isn't reported by anyone else is because it's reported to be rare. And because it's reported to be rare, people don't look for it. Now, we're fortunate in that Ancestry does look for it, but 23andMe, which 40% of our users use, doesn't properly and doesn't fully detect it at all. So if you've got the choice, I would always use Ancestry. Now, so this data only represents Ancestry data, i.e. 60% of our users. And you can see how it breaks down. But this breakdown is does not relate to the population as a whole, because as I said earlier, our users are mostly sufferers of hemochromatosis. They often know they're already suffering from it. So it's very biased in terms of sufferers, but also that's an advantage to us because we end up with more symptomatic people and we can analyze better. The other thing to say here is that we do have some type 2 people, which again is supposed to be rare, and in fact it is relatively rare for us as well. And the mutation of the type 1 gene, the HFE gene, called S65C, is also appearing in our data, but again mixed with one of the other types in some cases and on its own in others. Now this is also a very interesting graph because it shows the actual data users gave us for the age at which they got symptoms. And we've divided it up by the type of hemochromatosis that the user is suffering from genetically. Now, immediately you can see that the users get this disease at all sorts of ages, all the way from below 10 all the way up to 70 or 80. Now, if just looking at it like this, you'll think it's not age dependent because it's happening to different people at different ages. But I can assure you it is age dependent. It's just this is a cross section of people at different ages. And what we discovered was looking at individuals. You can actually say at any one age what their chances of getting symptoms are. So the next graph is our predicted age for the same people. And you can see that it's similar, it's not identical, and particularly not identical at the lower end of the ages. And we think the reason for this is we also discovered that some of the inflammatory diseases actually speed up the loading of iron if you're already loading or overloading iron. So conclusion is some of this reporting is from people, particularly again for this very interesting one, which is a carrier of H63D. That we've got people reporting that they got the disease at the age of 3 or, younger than 5. Now, what it is that they're actually reporting something else. They're reporting an inflammatory disease. But that inflammatory disease itself contributed to the extra loading of hemochromatosis and is the reason why a carrier who shouldn't particularly overload does overload. So it's a factor. It's a comorbidity in a way. It's two different diseases affecting each other. So this picture represents our findings in terms of what are the influences on the cell damage caused by hemochromatosis and other diseases we have. What we discovered from our analysis of the data using artificial intelligence was that other diseases such as glucose intolerance, lactose intolerance, the gut biome issues itself, such as IBS, and even a virus or a bacteria affect the immune system. And this immune system actually uses iron to control inflammation. It either converts pure iron to ferritin, which is a safer form and doesn't cause inflammation, or it gets macrophages to drop iron like bombs on things like viruses or bacteria to try and kill them. And in so doing, it actually causes inflammation. So what does inflammation do? It causes cell damage, and the cells end up being senescent cells, and these actually cause further inflammation. And the type of diseases caused by inflammation in different organs are heart disease, diabetes type 2, Alzheimer's in the brain, and Parkinson's disease, arthritis in the liver cirrhosis or liver cancer, and other diseases throughout the body. Now let's go back in history to Gregor Mendel, who discovered the modern genetics and the concept that for recessive genes, you need two genes to cause the feature. In his case, he was looking at pea plants and looking at smooth peas or wrinkled peas. The problem is this was an oversimplification, particularly when you transfer those ideas to the human body, where all the genes have a relationship with all the other genes, that genes do not act alone in normal circumstances. And this brings us to the concept of why doctors say that carriers cannot overload, because they're basing their ideas on an outdated, oversimplistic approach to genetics. Now, many people still say that iron overload is a monogenic disease. This just means it's based reliant on a single gene. In this case, they think it's only the HFE gene which is contributing to hemochromatosis. But I've already pointed out that 19% of our users have both type 3 gene, which is called TFR2, and the HFE gene involved. Also, we showed that even type 2, which is the HJV HAMP gene for juvenile hemochromatosis, is also involved. The other types, type 4 and 5, really are rare in our data, and in fact we haven't seen any. But as we said earlier, other diseases that cause inflammation also affect the rate of loading of hemochromatosis sufferers. We call this metabolic disease comorbidities. And such things as, in particular, lactose intolerance and other gut issues like IBS, gluten intolerance, diabetes type 2 and thyroid issues all seem to contribute to increasing the loading. And that's why we see carriers perhaps suffering from let's say lactose intolerance at an early age, then later on actually start suffering from hemochromatosis. But since the symptoms are almost the same, it's difficult for them to actually distinguish between the two. Let's summarize what our discoveries are from the CheckIron project. Firstly, we can do predictive DNA analysis to approximately calculate what age a particular individual will or may suffer from hemochromatosis. It's not perfectly accurate, and more data will help tremendously in honing this down. Secondly, iron overload in carriers really is possible. Perhaps the word carrier is not the correct word anymore. And in fact, one of the cases was that for one particular type of carrier, it's actually more common than what classic hemochromatosis is. Thirdly, type 3 hemochromatosis, which is supposed to be rare, is not rare and is nearly as common as type 1 hemochromatosis. And indeed, you can mix type 1 and type 3 at the same time, and this itself seems quite common. And fourthly, other genes do influence how iron overload happens in terms of changing the rate of loading. And finally, let's look at what we want to see in the future, and we'll be discussing some of this in the next video in a week's time. We want to see increased awareness of iron and iron overload. So a sizable proportion of the population does have too much iron, and it might be a similar amount to the number with too little. And we should be more aware of this because it alters the age at which we die quite significantly. Secondly, a near zero cost of DNA screening is now possible because millions of people, something like 26 million people in 2019, had already done consumer type DNA testing. And this means that the cost of predicting is a near zero cost of predicting hemochromatosis just from the DNA screening. Thirdly, we can do a comprehensive DNA analysis using things like CheckIron. And the fourth thing, which we will do a whole video about, is about rethinking about iron fortification in food. This is something that was started in the 1940s by Kellogg's and has gone out of control because today people with iron deficiency should be prescribed iron and not given it in food, where the dose cannot be controlled. It's as bad for those people without enough iron as well as being terrible for those with too much. Please like and subscribe to this video, and I look forward to talking to you more over the coming weeks on other subjects.